Now we said that the proteins are synthesized in the rough endoplasmic reticulum, and with the synthesis what happens that these proteins are packed into vesicles and they are targeted to another organelle, that is the Golgi apparatus or Golgi complex. This Golgi complex consists of membrane enclosed sacs, and which is facing, one side is facing towards the nucleus and the other side is facing away from the nucleus. Now this Golgi complex has lot of enzymes which can modify the proteins present in the vesicles. So these vesicles go and fuse with the Golgi complex, and here the proteins are concentrated, processed and packed. What does this mean is the major processing of the proteins as well as lipids take place in Golgi complex, that is they undergo proper glycosylation. So this Golgi complex has lot of enzymes that either add, remove or modify sugars from proteins and lipids. Then as it is being processed, it passes through the various sacs and finally the proteins are released in vesicles from the trans phase of the Golgi complex. So these vesicles are also labeled with an address. That means the targeting of the proteins, where they will go, whether they need to be exocytosed from the cell and released, or they have to be inserted on the cell membrane, or the proteins are targeted for the lysosomes, that address is given by the Golgi complex and the vesicle moves to the proper location so that proteins can go to their targeted place. Okay. Now let's move on to another organelle, that is peroxisomes. These peroxisomes are ovoid structures and they are seen in hepatocytes and proximal convoluted tubule in kidneys. Then they contain oxidative enzymes. These peroxisomes perform many functions, the crucial one being the synthesis and degradation of hydrogen peroxide. For this they have oxidases and catalases, and by virtue of this function they protect cell from reactive oxygen species. Then they also have role in synthesis of bile acids, as I have already told that they are seen in hepatocytes, then for cholesterol synthesis and also for fatty acid oxidation. Now let us see the cell organelle nucleus. Nucleus is the storehouse of genetic information. So what is the structure of nucleus? Basically this nucleus contains the genetic information, that is the DNA, which is condensed DNA, and that is known as chromatin. Now this is enveloped by double membrane, right? So these are the two membranes of the nucleus. In between the membrane, in between the outer and inner membrane, there is a space which is known as perinuclear cistern. Now this membrane you see is not continuous. There is some space in between the membrane. This is known as nuclear pore. This allows for movement of proteins and especially mRNA out of the nucleus. So you know that DNA is transcribed into RNA, and RNA is then translated to proteins by our endoplasmic reticulum. So that means mRNA should move out, isn't it? So they move out via this nuclear pore, and there are a lot of proteins which are present on this nuclear pore. Some of these are importins and exportins. They kind of form a channel here, network. They network and form a channel, right? So these are important for transport of some substances, especially of proteins and mRNA. So this chromatin, we said that it is basically has condensed DNA. Now nucleus also has a collection of granules known as nucleolus. So here this is the nucleolus. This nucleolus actually does not have a membrane. It is only a collection of granules and it is rich in RNA. So this nucleolus is the site of synthesis of ribosomes. So you know that ribosomes are important for the synthesis of proteins, isn't it? But for the synthesis of ribosomes, we need nucleolus. Okay, let's move on to the final component of the cell, that is the cytoskeletal element. Cytoskeletal element is basically a number of fibers that maintain the shape and structure of the cell. Not only they maintain the shape and structure, but they also allow the cell to change shape and move in case it is needed. So the cytoskeleton basically consists of microfilaments, intermediate filaments and microtubules. We will deal with them one by one. First, let's discuss microtubules. Microtubules are formed of a subunit which consists of two proteins, alpha tubulin and beta tubulin, which together form a dimer subunit. Now, these dimers come together and form a protofilament. So, here this marked you are seeing, it is forming a protofilament when these dimers are attaching to each other. 13 protofilaments come together, stack together to form a ring. So, here this diagram, if you see, it is showing the view from the front, and you see, if we count, it is 13 protofilaments. Now, this microtubule is basically in dynamic condition. That means it can grow from one end and it can also dissolve from other end. So, these actually units interact with the GTP, and where these subunits can bind, that is the plus end, and from where these subunits can release from the microtubule, that is the negative end. So, in this case what we are seeing is microtubule is growing in this end, and dissolution is taking place from the negative ends where this dimer subunit is detaching from the microtubules. So, because of this dynamic nature, microtubules act as tracks on which movement of substances can take place, carrying substances from one place of the cytoplasm to the other place. So, here it is shown vesicular transport, where certain proteins known as molecular motors transport substances on the microtubules. So, basically these proteins are walking on the microtubules. So, when the transport takes place from negative end of the microtubule towards the positive end of the microtubule, that is known as anterograde transport, and it is brought about by molecular motor known as kinesin. On the other hand, when the transport is occurring from the positive end towards the negative end, that is known as a retrograde transport and is brought about by molecular motor dynein. So, we have seen in Golgi complex when we were talking about that, how the proteins are labeled and are carried to different addresses. So, they walk on these microtubules via the molecular motors and they are carried to different places. Then microtubules are also responsible for the formation of the mitotic spindle. So, here this is microtubules, and this is the organizing center for microtubules from which the microtubules are forming, and the chromosomes separate on these tracks formed by the microtubules, that is the mitotic spindle. Now, in the previous slide we said that microtubules are arising from microtubule organizing centers. So, what are these? These are basically formed from centrosomes, which is made up of two centrioles. Now, these centrioles are present near the nucleus. So, microtubules arise from these centrioles, and these microtubules are arranged in stacks of 3. So, you see these 3 stacks here. So, this is stack of the microtubules which is arising from the centrioles, and together they are arranged as 9 triplets. So, if you count, it will be 9 triplets of the microtubules which are running at a little angle from each other. So this centrosome acts as microtubule organizing centers, and they have another protein, that is gamma tubulin, and it is from these gamma tubulin that microtubules can basically aggregate, the subunits of microtubules can aggregate from this gamma tubulin and form the microtubule filaments. Now, apart from the vesicular transport and formation of mitotic spindle, microtubules are also responsible for formation of cilia and flagellum. Cilia are specialized structures which are responsible for moving substances over the epithelia. For example, in respiratory tract, mucus is moved over the epithelia and it is moved through the nose, then it is then sneezed out. So, how these cilia function? Basically, you see, cilia consist of two parts. This yellow part shown is axoneme and there is basal body. Basal body is same as the centrioles which we saw, that it is consisting of triplets of microtubules that are arranged, 9 triplets are arranged in a ring form. So, that is the basal body, similar to that of the centrioles. And axoneme consists of microtubule doublets, not triplets, doublets. So, here 9 microtubule doublets are there, and then there is a central one doublet also. So, basically two inner microtubules are there. So, this is known as 9 plus 2 arrangement of the microtubules. Then there is interaction of the protein dynein, which we also saw in the vesicular transport. These interact with these microtubule doublets, and it is the coordinated interaction of these dynein and microtubule which is responsible for the movement of the cilia. Okay, let's move on to next cytoskeleton element, that is the microfilaments. Microfilaments, just like the microtubules, also consist of subunits. So, these are monomer subunits. In microtubules, we saw as dimer subunits. So, these monomer subunits are globular actin or G-actin, which interact with each other and form the filamentous actin, that is the F-actin. Now, this filament, just like that of the microtubules, also can form at one end and there can be dissolution at the other end. So, this is responsible for muscle contraction, that actin myosin interaction which occurs in muscles is responsible for muscle contraction. Then it also by interaction with the integrin receptors present on the membrane, it forms the focal adhesions, and it is also responsible for adhesion belt by which the cells attach to each other. Then it acts as tracks for molecular motors, just like the microtubules. Plus, it also forms microvilli and lamellopodia. Lamellopodia is responsible for cell movement. So, when the cell is moving from one place to other place, these filamentous actin actually form on the leading edge of the cell. So, where it is moving, filamentous actin keeps on forming and there is dissolution from the other end. Okay, coming to the final cytoskeletal element, that is the intermediate filament. Intermediate filament are more or less static and they provide structural support to the cell. So, if there is a problem in the intermediate filaments, then the cells rupture more easily, and it has been found that in certain conditions that there may be blistering of the skin if these intermediate filaments are abnormal. Plus, the protein subunits which form the intermediate filaments, they are very cell specific. For example, in fibroblast, vimentin is the major intermediate filament, which is vimentin forms the major intermediate filament, whereas in epithelial cells we see cytokeratin. So, with this we have discussed about the complex machinery of the cell, how various organelles are responsible for various functions of the cell. Some is acting as synthetic machinery, some are acting as excretory machinery, and then there are cytoskeletal elements which are providing the structural support to the cell, forming tracks and helping in its mobility. Thank you. Have a nice day.